Good morning to everybody. So the question for everyone who is working in the field of human neurobiology is really what makes us human. But my specific concern is how this human-specific circuitry and connectivity emerges during development. Here we, on the first place, have an internal representation, flexible behavior, and really special abilities like culture, and including thinking about God and religion. It's a very complicated issue, and it's very challenging for you people in the field of artificial intelligence, because really to reconstruct all these events, namely we have here a lysencephalic brain, and finally will be a brain of adult person with gyri. Then you have here in the fetus just few regions, region typical areas, but in adult you have parcellation, a very complicated one, and nobody agrees how it should be parcellated. Then in a fetus, you have, let's say, few layers, but they are completely different than these 6 adult layers, and that's a crucial thing to remember. Then we have fetal columns, and we don't know the relationship with adult columns, because the modular columnar organization is a crucial aspect of adult organization. Of course, the issue is how simple circuitry with few synapses develop to beautiful connectoma, you know from your graph analysis and imaging studies. And here I point, this arrow points to the pyramidal neurons, very immature. That's original Golgi. Nobody has so early Golgi or pyramidal human neurons. And then we come to adult circuitry, which is also centered around the pyramidal neurons together with the interneurons. Of course, synapses are a special challenging problem. You have few synapses. In adult, you have 10,000 times 28 billions in 3-dimensional architecture. And for presentation, you can find on pages of this meeting. I listed what features we should study in fetus and how it's different from adult. And this red arrow means that in between you have enormous reorganization and development during late preterm period, infancy, childhood, and adolescence. And what is white? White is white areas mean that we cannot approach with magnetic resonance, and artificial intelligence has no approach to these issues. And the questions in this context of development of circuitry are really when something appears there, that's a very crucial parameter. What type of circuitry we have, endogenous or externally driven, and really how afferent pathways developed. It's a complicated system of afferents and efferents, and how they meet the postsynaptic elements. So Professor Judas presented postnatal development, and in this presentation I will concentrate on early fetal development, because there are no magnetic resonance studies, only anatomical study from this period, and some physiological studies, mostly correlated with animal corresponding phases. This diagram shows how intensive are different processes in different periods, from proliferation, which means production of neurons, migration, molecular specification, neural aggregation and cytoarchitecture, axonal ingrowth, and finally with pruning of exuberant connectivity. But neurochemical maturation, myelination, and synaptogenesis rearrangement is practically lifetime process. So I was asked by Catherine to pay a little bit more attention to the transient compartments of the cerebral wall in the developing mammalian brain. I will talk only about human, but I will use evidence that the oldest neurons are born early and they occupy deep layers, and the youngest neurons finish here in superficial associative layers. This is from Paško Rakić study when he dated every class of neurons in the developing monkey brain. And later we will come back to these layers. This is up-to-date scheme from one symposium in Oxford. You see, here is a proliferative zones, ventricular, subventricular. Here is intermediate zone with axons. Then you have a transient compartment subplate, which is very important for me. I will tell you why. And then we have a cortical plate with first with projection neurons and then with associative neurons. And on the right, there are listed processes, neurogenetic processes, proliferation, migration, differentiation, synaptogenesis, axonal outgrowth, and cell death. So that should be reconstructed in time and in space. Of course, my concentration is here on the so-called synapse-rich compartment. That means this green is just producing neurons. Then they migrate and fibers go through the intermediate zone, but then this layer, subplate and marginal zone, they receive first synapses. So for months and exactly until 22 weeks of gestation, so for several months, cortical plate, I mean the area where this CP with black cells is without synapses. So synapses are concentrated where apical dendrites of neurons receive axons, and in this enormous connectivity layer subplate. So now that shows how these connectivity layers grow above and below cortical plate. And on the right is something which was very exciting for me, and I didn't see that other people are so excited as I am. Look, the largest compartment with synapses, you can see it macroscopically on conventional magnetic resonance imaging. So all layers can be seen in conventional magnetic resonance, starting from this ventricular, subventricular zone, then intermediate, and then huge subplate, and then cortical plate and marginal zone. The problem is that the major input to apical dendrites arrives in marginal zone, and that zone is not seen in any, even the highest resolution magnetic devices like 7 Tesla, even 9 Tesla, whatever. So the crucial point is that you see the major synaptic compartment on conventional magnetic resonance. You can measure volume, you can see whether it's normal. Why you see it? Because it has water content. We will come to this later. And now, step by step, I will go with you from early fetal to mid-fetal, and then we will finish with perinatal reorganization. Why I like this? Because I spent 2 years counting synapses with such a beautiful vertical probes on Philip 200 microscope. This is fantastic fixation, and we plotted the synapses. You see how they are distributed in layers. So absolutely selective sub-layer is synaptic reach. And here is from publication with, there are 2 young ladies, it's under review in Cells, to show that during early organization, this is a marker of deep projection neurons. The whole cortex is composed of cortical plate, densely packed, deep projection neurons. That marker is TBR. So it's a marker of deep projection neurons. Here is are neurons, some migrating, and some neurons like Hall-Reitz cells are in marginal zone. And this is the earliest circuitry. You know, it's amazing that, like in adult, this afferents and efferents originate and terminate in layers. This time in transient layers. For example, pay attention to this, the earliest input from tegmentum, it's a modulatory, it goes to the marginal zone and to pre-subplate area. So this is the earliest circuitry. It is endogenous, it is not sensory driven, and it is self-organized. That's crucial. We call this this activity mostly oscillatory. I will show, come to you later, but now it's one event which you don't see in the rodent, and it's modest in carnivora, but it's really the most developed in human and monkey. And this is that the deep part of cortical plate simply enlarges, expands. Here is 1 micron section. You see, even on such a thin section, plastic, you see this spread down, and here are the synapses. So despite this enormous spread down of subplate, expansion of subplate, which is characteristic for human, still the synapses are in 1 layer above, and now in subplate below. And what is amazing, if you look so far, I think that's the earliest in vivo magnetic resonance I have seen in the literature. You see, I see this second plate here. See, even in utero with conventional magnetic resonance with correction of movement and so on. So you see even this event, formation of synapse-rich layer. The question is how the experimental people who were doing this, like a group of Professor Luhmann, but before Carla Schatz and her group, they simply confirm that, not simply, it was very complicated to record from immature cortex, but they confirmed that action potentials are located in subplate, and that cortical plate, which has these embryonic columns, is devoid of action potentials, and you have oscillatory activity with some hints of clustering. And in this area, I can show very beautifully early synaptic contacts. So electron microscopy correlates well with the recording from these layers. Now it's really astonishing that something in the middle of fetal life, even earlier, around the 13, 15 weeks, you have most synapses, or the most numerous synapses are in hippocampus. So there is no memory at that time. Nobody remembers what was going on in the first year, even second year of life, but synapses are there. And the group in Marseille showed in monkey that at that time you already have action potentials and activity of pyramidal neurons. This is also from submitted manuscript. Here, the same thing is in singular cortex, which you see here are also markers of deep projection neurons. That's a limbus, the end of hemisphere. And here I counted synapses, and you see that the majority of synapses, that's graph, are in superficial compartment called marginal zone. So what you should remember, the limbic cortex is special, that it has early development, and ask yourself what are synapses doing there before the memory. I will come to this later. I think that episodic memory already happened at newborn stage, and I mean at the birth in newborn babies, and therefore you have hippocampus. But complex, long-lasting memories are related to associative cortex hippocampal interaction, and that does not occur before 2 years of age. Now dendrites. I figure out by reading papers from your field that dendrites are important. Of course, for me, dendrites are crucial. Why? Because you see, in subplate, for example, first synapses are here, an apical proximal dendrites. If I go in marginal zone, see, this is a real Golgi reconstruction of pyramidal neurons. And here is a beautiful synapson bifurcation of apical dendrites. You see, this early arriving axon makes synapse on both sides of this apical, and that we don't make a fantasy. These are real cells from this early fetal age. Now is the question, how about interneurons? When I started to work on developmental human neurobiology, that was a long time ago. You have not been born at that time. At that time was a rule based on studies in rodents. First, big neurons are born, then small neurons are born, and then glia is born. And that's not true for the primate brain. There is a parallel development of pyramidal neurons, projection neurons, interneurons, and glia. Just to convince you, here is a beautiful interneuron in mid-fetal age, a double bouquet cell, one of the very important neurons. And here is distribution of, let's say, calretinin neurons. And you see that they are concentrated here where the subplate is being formed, what is here shown as deep subplate, this connectivity layer. Now, one thing is how I know that the pathways come there and that they, from where they come. It seems that it is impossible to do in human, but it is possible because you simply have to follow from some marker. You can use synaptophysin SNAP25, or I used in the beginning cholinesterase. So it is first you see the reactivity in thalamus, then you see reactivity in capsula interna and thalamocortical radiation, then you see activity in the subplate, and finally you see activity in cortex. So we know from where and how fibers are distributed. Here on the right side, you see this beautiful growth cone, how it grows, and using a gradient of guidance molecules. These gradients are very crucial. You see, for example, here a gradient is high, and this axons is using this gradient. You can, by deep learning, you can calculate these gradients and see how gradients should be arranged that axon finds address. Don't forget that that's already 1 centimeter of axon has to pass. And in let's say basketball player, can you imagine how axon from pyramidal neurons has to reach the spinal cord? So it's really a rigid, genetically controlled system of wiring. And to convince you that beautiful demonstration is possible, I show this. You see, it's a thalamus. Here is the activity. Then you have a thalamic radiation, and the fibers come intermediate zone, leaving cortical plate and telencephalon without activity. On the right side is one of the most important modulatory pathways in the brain, originating from cholinergic basal forebrain via capsula externa, and again not reaching at that time cortical plate by going into intermediate zone. So this picture is like something artificial, but it's a real thing. So you can see how wiring has been formed in fetal life from this major projection subcortical nuclei, thalamus and nucleus basalis. And now we come to the thing you hardly can imagine, and the people who are working on development or connectivity, simply they ignore. And this is that we have in these synaptic layers enormous extracellular space. And there are papers showing that in this extracellular matrix, you can have diffusion of active molecules, even transmitters like glutamate or other growth active and signaling molecules, which can allow communication between cells before synapses were formed. So again, show the enormous subplate, and it's full of this. We will come to this problem later. Now for people who are doing reconstruction of pathways and so-called geometry of the white matter in telencephalon. I noticed that they don't see the major difference between neocortical afferent system, that's DTI, reconstruction of thalamocortical radiation, or sagittal strata, visual pathways from deep visual centers and pulvinar coming to the visual cortex. But on the right, you see, that's on material of Susumu Mori from Hopkins and Dr. Huang. You see that the limbic pathways, they keep together, they form bundles. So there is something on limbic cortex, that is cingulate cortex, parahippocampal cortex, and hippocampus, what makes these bundles keep together, so they are real bundles. And another point, almost forgotten, Dr. Hevner and Kinney, they have shown that human hippocampal formation, if traced by DIE, shows early intrinsic connectivity. So that confirms my hypothesis that the circuitry of hippocampal formation is ready to act at birth. But what is doing, we still just make hypothesize. Now to the subplate. Why I like subplate? Not because I described with Dr. Molver first subplate. I like subplate because you can see it in vivo magnet, then in postmortem with Nissel as the largest compartment of human fetal brain. And now to repeat the layers, there is a ventricular zone producing neurons, then internal subventricular zone also producing neurons, then enormous external subventricular zone mixed with fibers which make here a sagittal strata. Then we come to the subplate, then is cortical plate, then is a transient subpial granular layer within this marginal zone. As I said, nobody can show this on imaging analysis. And subplate shown here with blue, brown, yellow, and synaptic distribution. It's really can be considered as a waiting compartment. You see the axons come, they find their partners, but that's just a step before they grow into cortical plate. But what is the real substrate of organization of subplate? You see, it's a kind of a random network, a really maturational network, which does not exist in adult in this shape, and it serves for transitional period before you have domain-specific activity, which is important for preterm and newborn learning. What I mentioned in this panel discussion, you see early development of beautiful astrocytes, isodendritic core and polymorph neurons, and evidence that they are postsynaptic, I find here. You see, that's a subplate neuron 5 millimeters deep. That's thicker than the adult cortex. And here you see clear synapses on soma. But I must say, most of the synapses are axodendritic and are excitatory. So even so-called GABA is maybe excitatory in developing brain. So now we learned that you have a random network, but it's crucial. It goes from limbus to basal aspect of hemisphere. So it's a global network which covers the whole developing brain. Now, point important for neurophysiology and this reconstruction of connectoma and circuitry. Here I point just one thing, that this is a work of Carla Schatz from subcortical visual cortex. Axons are coming to subplate. You put electrode, you stimulate the visual system, you get response. But in the same time, and that's in human only after 24 weeks of gestation, you get response in cortex. So subplate neurons are not only important as early postsynaptic elements making a special tangential nexus. They're important as amplifiers of thalamocortical activity. You see, that is shown in our schematic drawing. That's from paper with Judas. We put here this white spot is actually a layer 4 neuron, which receives input from thalamus and from subplate. So amplifier of thalamocortical domain-specific circuit. Now, this is, I forgot that I have arrows to these points. Now, this layer, you can reconstruct volume, and this is from the joint paper with McGill. Lana Vasung did it for doctoral thesis. You see that this red area around the Sylvian fissure, it's actually shows that subplate is thickest in associative areas. That means after finishing with thalamocortical circuitry, subplate is involved in cortical circuitry. And that was measured by methodology from McGill, and I'm happy that we did it. But now that's reconstruction of this circuitry. You can find in a review article I wrote specially on subplate nexus. But the point is, look, during development, there are multiple, that's crucial, multiple inputs from associative fibers and thalamocorticals. You see this blue shows multiple inputs, now arrows. So that's crucial. The one axon coming from deep nuclei within the subplate makes inputs to several places and actually integrates the large areas of cortex. So this is a special thing in development. In adult, there are some thalamic nuclei which project not only in specific area, but Klaska, Dr. Klaska have shown this in several papers. And here I'm happy that one of our former students, now professor in Rutgers, Rocco Rashin, he has shown that there is a special clustering of genes in subplate. So this circuitry has a special clustering of genes. Why? Because the synapses are there, 100 proteins should be formed. Then extracellular space is there with guidance molecules. So again, you need special transcription factors. So subplate is not only special for connectivity, it has also a molecular specificity expressed with clustering of genes, which is shown on the right portion. What was my point with this mark? That within the subplate, there is chain of neurons. What means chain? You can follow this chain, look, through the capsula interna to the brainstem. So this is a special system of isodendritic core neurons described by Russian authors. Nobody pay attention to this. Judas is studying this, Sedmak is studying this as interstitial neurons, but I'm now talking that there is continuity from the brainstem to the subplate of this chain of neurons. We have shown this in the first paper with Paško Rakić, reported to this Ramon Moliner paper and Russian authors Zhukova, because I think that's extra to the reticular activation system. In addition to the modulatory system, it's a chain of neurons, a very ancient, probably from reptilian period. And now it comes something for clinical science and therefore for you as imaging and artificial intelligence people, important period. And this is after 20 weeks of post-conceptual, let's say gestation. It's 15 weeks different, 15 days difference. At that time, the human preterm is viable, and it's when it's born, it's a citizen of Croatia or United States or Germany and so on. So it's a real citizen. It gets name, and you like that it survives even if it's so tiny and fragile. And at that time, we have found first synapses within the cortical plate. Look at synapse. There is no extracellular space. Here is schematical drawing synapse, this little branching, and here synapses in the cortical plate. And then later, nice studies of Swedish group and London group, Mary Fitzgerald, they have shown that you can evoke at that period, after pain stimulation, you can evoke cortex. So the learning is possible by external input. Because here it's a mother voice, phonemes, and response in auditory cortex. It's a wider area, but it's a response. So that begins interaction with environment, with visual stimuli, if the preterm is born. Of course, in uterus, we don't know what kind of interaction, but if you make a tremendous sound, will also react the fetus in uterus. But here I'm talking about visual, auditory, and somatosensory stimuli. We are good in time, I hope so. Thank you. And now, enigmatic question. When associative fibers develop? Associative fibers means they originate from layer 3, pyramidal associative or mental neurons, and they make a long connectivity. So by definition of neuroanatomy and combination with modern language of connectomics, they make a long range part of connectoma. And these pathways develop in utero at the end of gestation. But in the beginning, after, let's say, around 24 weeks, they didn't yet penetrate the cortical plate. Look here. That's a study with Hardy method, Wasung, Takahashi at Harvard. I'm also co-author. Look, this is an associative bundle, and it reaches this arrow point, didn't enter cortex. It's still growing below subplate. And this is my explanation for several studies of Moria Thomison and this group, when they show in preterm infant high order areas connectivity, let's say here between future Broca area and parietal cortex. I think this weak activity is through the subplate, not through the real synaptic engagement of long cortical pathways. That happened perinatally and postnatally because the synaptogenesis is predominantly postnatal event. First synapses are crucial that neurons recognize that they make an outline architecture, what you call architecture of network, but the synaptogenesis is postnatal event. And just to show that during this period of reorganization, which is actually perinatal event and early postnatal event, is tremendous reorganization. Pathways being retracted, some enter the target, synapses develop with explosion. It's really a challenging, fast process. at that period, you still have subplate remnant. I think that Professor Milan Radoš will tell something about this. And this is just to show in monkey in visual cortex of rather old fetus that there are cells and with synapses in so-called subplate remnant. This is a conclusion. You can read from my power presentation, you can find online, but I just point here, not to take too much time because time is dashing by, that innate connectivity precedes domain-specific activity. And second thing, all neurons are born and all long connections are there. So a newborn, in contrast to the preterm infant, is ready to accept multimodal inputs. It's able to learning, especially in the field of emotional behavior and allostatic, reaching allostatic goals to get food, to get contact with the caregiver, and to be safe. Actually, this emotional, visceral behavior is already there. And this old scheme, but nicely shows the battle between scientists, what is more important, genes or environment. Of course, both are important because environment shapes on genetically based wiring and so on. And if we have this problem, what's happened during early postnatal period when the infant starts to learn? It has a fast learning, one-shot learning, unsupervised learning. So you should ask yourself, can I do something in artificial networks which is close to this fascinating fast learning of a human infant? I just here show that there is a continuation of subplate development dendrites, and here are cells, interstitial neurons, which originate from the remnant of subplate, but altogether saying that during the first year, you still have transient connectivity, but the majority of event is really, that is here, you see this blue line, SEDMAC help with this scheme actually, and you can see in conventional magnet, and I point, and here I don't agree with the connectomic people. The short cortical connectivity continues in human cortex after birth, especially U fibers. And it was shown many years ago by eye tracing by Dr. Burkhalter. So I think that scheme, small wilderness, then long connection, short connection, then long connection is not completely correct, because you see predominant event, but you don't see the fine. Okay. And just to show that really during first year, this is from the recent paper in Neuron, we publish in cooperation with people from St. Louis. You see our histology of this border between cortex and white matter, this not finished border. You remember, Alan, you told me, that I have problem with cortical thickness if I do this in the first postnatal months. And that's because you here have no sharp border, and we have shown, actually not we, they have these tools, they have showed this changing border between white matter and cortex with this quantitative methodology of imaging. And now reorganization. I don't know how the various procedures, how you cover, but my point is now that during unsupervised learning, one-shot learning, and everything what happened in postnatal time, you have explosion of synapses. That means that if you follow, that means explosion. So they reach the peak in associative cortex around the 2 years, and in visual primary cortex around the first year of life. So parallel to this, there is a development of dendrites. Of course, first pyramidal neurons or projection neurons, and then associative a little bit behind and reach the peak later. And during this period, all major networks are revealed by resting state activity are there. But for me, the fascinating thing is how consciousness emerges. And in this context, it's important that two groups have shown that the earliest, one of the earliest activities by resting state in a newborn is in medial prefrontal area, including anterior cingulate cortex and precuneus retrosplenial area, which are known hypothetically as self-awareness and theory of mind areas. The question is whether the people have shown this reorganization in network. For example, paper of Gao, yes, he showed. Here you have an immature network. Then in neonates, you have some kind of over connectivity and then diminish. So axons retract, they are being pruned, and Andrea told me that there is expression drop out. That's a normal thing in callosal projection. Half of the neurons have a dropout. In visual primary system, also 1/3 of all projection fibers. Probably that's in all systems. And this is not a clear plasticity. It is actually overgrowth, pruning, and kind of a regressive process. Now, the question was why this baby with 9 months can have such a triadic behavior. Is the distinguished examiner, object, and himself. The difference between this baby's network and your artificial system is that baby has a multimodal simultaneous input: touch, smell, movement, vision, everything together on already formed network. That's crucial for this. And just is a summary. I need one presentation for this, but will not do. And I was happy to see by the doctoral thesis of Klaus Hilgetag that he was one of the first artificial intelligence people showing that for network is a crucial, for a real world network is a crucial space constraint. That means when you measure this clustering coefficient, like here, and average short pathways, it's in a limited space, contrary to adult, where you have many skull-free networking. And Catherine asked me, show example of early one-shot learning. You see, I think that's maybe not one shot, but almost one shot. Look, the circuitry is there by birth. Amygdala, orbital cortex for reward, subcortical visual center, of course, and early input to fusiform gyrus, and eventually, although I don't believe completely to this paper, a baby can definitely recognize the face, not mother's face at the birth, but face as a object, and can eventually imitate, you know. So we said, whom is looking that baby, to whom is showing this. But anyhow, this example, because this baby sucks, not because muscles are ready, because it feels, the mother feels the touch, it has an oxytocin binding, and it's really complex behavioral learning. It learns extremely fast because it's necessary for survival. Okay, and now that's our institute, and people who I have collaborate, everybody is here, and these two ladies are in red because they helped me with the presentation. That's it.